This is a review of antibiotics for EMS providers. Antibiotics are a subgroup of a group of compounds called antimicrobials. Antimicrobials are substances that affect the growth or the ability of certain pathogens to survive. Antimicrobials can be either naturally occurring or they can be synthesized in the labs or modified from naturally occurring substances. Several terms we're going to talk about today are listed here. Acquired resistance is basically the ability of a pathogen to actually develop resistance to a certain medication or antimicrobial medication to use against it. Antibiotics are a type of antimicrobial that fight bacteria. They are considered antibacterial drugs, as opposed to antiviral drugs, which affect viruses. We'll talk about those in a minute. Antibiotics can be either broad spectrum or narrow spectrum, depending on whether they affect a wide variety of bacteria or very few bacteria. Pathogens themselves can be of many different types. One type of pathogen that we're going to talk about today are bacteria. Bacteria are single-celled organisms that do not have an organized nucleus. They have DNA, but that DNA is not incorporated in a double-membraned nucleus like ours are. That's to be compared with viruses, which are actually just fragments of either DNA or RNA encapsulated by protein. They are not bacteria. Fungi, which are also simple organisms that can be either unicellular or multicellular, such as mushrooms and yeasts, and then protozoans, which are also single-celled organisms, but protozoans actually have an organized nucleus enclosed in a double membrane like ours. Bacteria themselves can be classified two different ways. One way to classify bacteria is by the shape of the bacteria. Bacteria can be either rod-shaped or rectangular in nature, such as Escherichia coli or E. coli. They can be spiral-shaped. They can be somewhat twisted in shape, such as Treponema palladium, which is the spirochete that causes the disease syphilis, or that can be ball-like spheric structures or cocci. Most of us are familiar with Staphylococcus or Streptococcus. Irregardless, they all have the same other properties that antibiotics have that we talk about. The other way you can classify antibiotics is whether or not they are what are called gram-negative or gram-positive. Gram-negative and gram-positive bacteria are based on the ability of the bacteria to be stained by what's called the Gram stain, which was devised by a Danish bacteriologist named Hans Christian Gram, who noticed in 1884, while he was working in the morgue, that he could identify some types of bacteria from others, whether or not they picked up what's called the Gram stain. The Gram stain or not having picked up the Gram stain. So some bacteria are gram-positive, they stain bluish. Some bacteria are gram-negative, they pick up the Gram stain and are shown here in red. So antimicrobials are a group of naturally or synthesized substances that kill microbes, and antibiotics are a subset of those. These are a subset that are effective against bacteria. Antibiotics can be naturally synthesized, or they can be synthesized in the lab. Irregardless, their job is to affect the growth of or kill bacteria. The first antibiotic was produced by a man named Alexander Fleming. He was a Scottish microbiologist, and in 1928, in August, he took a vacation with his family, and at that time he had been growing plates of Staphylococcus. He left these plates in his lab, came back in September of that year, and realized that some of his plates were contaminated. And you can see here's an example of his plate, and at the top, that large white blob is a contaminant that was growing on that plate, and he found that that was a mold called Penicillium, which is a mold. And he noticed that around where that mold was growing on the plate, there were no colonies of Staphylococcus nearby, to which he supposedly historically pronounced, "That's funny." And he decided to look at what was going on that did not allow the Staphylococcus to grow near the Penicillium mold. His study found that the mold was actually producing a substance that was inhibiting the growth of the Staphylococcus. He named that substance penicillin, and penicillin was then discovered in 1928. It was the first substance that we had to treat bacterial infections, and it was widely used to treat many, many different infections that up until that point we had no way of treating, leading to advertisements such as this one, allowing penicillin to treat a problem that had not been able to be treated in the past. The penicillin that Alexander Fleming, who by the way won the Nobel Prize in 1945 for this, was called penicillin G, or benzyl penicillin. It looks like this, and it is now today considered a narrow spectrum bacteria because of the fact that over the years bacteria have grown largely resistant to this. It is still sensitive to a number of infections. However, there are many types of bacteria that are not susceptible to this antibiotic anymore, and so it is considered what we call a narrow spectrum antibiotic. It has a complicated chemical structure, as noted. We'll talk more about that in a minute. Broad spectrum antibiotics have been developed which are effective against wide ranges of bacteria. Again, most of these are synthesized. Many of them are synthesized from the penicillin that Alexander Fleming had devised in the past. We'll talk about these as we go. The list here does not include all of the antibiotics, but it does include antibiotics that are commonly used nowadays to treat severe infections, sepsis, and things that may be involved in patients during EMS transport. We've listed them here as the trade name first and then the generic name in parentheses. How do they work? Well, if you're going to devise a substance or a naturally occurring substance going to be devised to fight bacteria, they have to be able to fight the bacteria and attack the bacteria without attacking our own cells. Bacteria, as we said before, are considered what we call prokaryotic cells. Pro meaning before, karyo meaning nucleus. The prokaryotic cells mean that the DNA of the bacteria is not enclosed in a membrane. That's one difference. Bacteria have several differences between their structures and our cells, and those are the sites in which antibiotics function to kill the bacteria but not kill our living cells. This is a schematic of a diagram, a generalized bacteria, and again, what it has in it is it has an inner membrane, which is shown in black. The cytoplasm includes the DNA, which is enclosed in one single circular chromosome. Outside of the cell membrane, there is a cell wall. The cell wall is something that bacterial cells have that we don't have, and that's going to be one of the ways in which many bacteria are attacked by antibiotics because antibiotics will not affect our cells since we do not have cell walls. We also do not have one single chromosome of closed circular DNA. We have our nucleus enclosed in an envelope. That's another site in which bacteria differ from our cells. In addition to that, in the cytoplasm of the bacteria consist structures called ribosomes. Ribosomes are the organelles of the bacteria that actually take the information that's delivered from the DNA and make proteins. We have ribosomes also, but as we'll see, our ribosomes are different than the ribosomes of the bacterial cell, and that provides another site in which we can attack bacteria without attacking our own cells. The bacterial cell wall, as I said, is something our cells do not have. The inner cell membrane or plasma membrane is all we have around our cells. Outside of that, bacteria have a cell wall, and depending on whether it's a gram-positive bacteria, as shown here on the left, or a gram-negative bacteria on the right, you basically have a cell wall consist of a protein and carbohydrate layer called peptidoglycan. And then in a gram-negative bacteria, in addition to that, you have another outer membrane outside of that. These are things that our cells do not have. We just have the plasma membrane. In addition, bacterial DNA is different than our DNA. Bacterial DNA consists of one closed circular loop of DNA, but bacteria also have circular loops of DNA that are not associated with bacterial DNA that are called plasmids. Plasmids convey certain genetic abilities to the bacteria. In many bacteria, plasmids can include directions for coding for proteins to allow for antibiotic resistance. Our cells largely do not have plasmids. Plasmids are also able to reproduce and also able to be transferred from bacteria to bacteria as the bacteria divide. Again, we said that both our cells and bacterial cells have to make proteins, and they do so utilizing organelles called ribosomes. Ribosomes exist in the cytoplasm of both our cells and the bacteria. They take direction from the DNA and they produce proteins, allowing our cells and bacterial cells to do what they do. The difference is that our bacterial ribosomes, our ribosomes and bacterial ribosomes are different. Bacterial ribosomes consist of two components, as seen in the top half of this chart, what's called a 50S and a 30S component. These components are made of ribosomal nucleic acid, or RNA, and these are the structural components of bacterial ribosomes. Our ribosomes, mammalian ribosomes, consist of 40 and 60S RNA components, and so you have a difference again between bacterial and our cells in the ribosomal protein synthetic mechanism, and it provides for a source where we can devise substances that can affect bacterial ribosomes but not our ribosomes. Folic acid, or tetrahydrofolate, is a substance that's needed both by us and by bacteria. Tetrahydrofolate is important, as you can see in this chart on the left, for producing what are called purine nucleotides, which are structural components of both DNA and RNA, as well as protein components of the cell when the cell makes certain proteins and needs specific amino acids. Both our cells and bacterial cells need folic acid. The difference is we can ingest folic acid in our diet, ingest tetrahydrofolate, and make these. Bacteria cannot. Bacteria, as you can see way on the right, on the left of this chart, have to actually begin with primitive components, as shown here, and actually produce the folic acid or the tetrahydrofolate. Because of the bacteria's need to produce folic acid and produce it from these precursors, that enables us to have other ways in which we can attack the bacteria with medications, antibiotics, and not attack ourselves. So as we go through talking about the types of antibiotic actions, we're going to use this chart as showing a typical bacterial cell and showing ways in which certain antibiotics act to disrupt the bacteria, but yet not disrupt our cells as we take it. The first way in which many bacteria are disrupted by antibiotics is by inhibiting the synthesis or the production or the development of the cell wall. Again, our cells don't have cell walls, so we don't have anything for these antibiotics that could disrupt. And as we talk about these, there's three major groups of antibiotics that affect cell wall synthesis. First being a group of medications called beta-lactams. Beta-lactams are a group of medicines that includes penicillin, as we initially talked about. Penicillin and the beta-lactams all have one thing in common, and it's shown here circled. This is called a beta-lactam ring. This is a chemical ring produced with 3 carbons and a nitrogen, and this beta-lactam ring is found in all of the beta-lactams. These beta-lactam antibiotics, because of this beta-lactam ring, are able to disrupt the cell wall of the bacteria. Thus, as the bacteria tries to grow, the wall disrupts and the bacteria dies. Penicillin G, as devised by or as discovered by Alexander Fleming, is shown on the top here with its beta-lactam ring. Other penicillins or penicillin derivatives are shown below it. These all consist of variations of the initial penicillin synthesized in the lab. At the bottom you'll see ampicillin. Basically it consists of the same basic structure as shown above, but the side chain has, instead of a CH2 group and then the ring, it has a amine group on the bottom that gives it more broadened coverage against bacteria that may be resistant to the basic penicillin G shown at the top. The important thing is that all of the beta-lactams have the beta-lactam ring in them, and that the differences are only in the side chains associated with it. So antibiotics such as penicillin kill bacteria by inhibiting or interfering with the production of the cell wall, so that when penicillin is in place and the bacteria, in this case E. coli, start to grow, the cell wall is weakened, it ruptures, and when the bacterial cell wall is ruptured, the bacteria dies. This is a schematic diagram showing the cell wall of a bacteria. The greenish structures are the cell membranes with a large brown protein going through it, and then above that are the carbohydrate molecules that make up the portion of the cell wall, and then the protein connections between them are shown there. At the top you can see the beta-lactam drugs affect those proteins that attach to the carbohydrates that make up the peptidoglycan cell wall, and when these beta-lactam drugs are in place, these connections are disrupted, so that as the bacteria grows, the cell wall ruptures and the bacteria dies. This is a list of the beta-lactam drugs that we're going to talk about today. Again, this isn't all of them, but these are lists of commonly used beta-lactam drugs used in serious, severe infections such as sepsis. And we're going to talk about them by classes, penicillin being the one we've already mentioned. We will talk about cephalosporins next, then the carbapenems, and then finally the combination drugs, which consist of two drugs. But all of these do the same thing. They all act to disrupt the cell wall of the bacteria. They're just made and synthesized so that they can affect more resistant bacteria. They all have the beta-lactam ring. The cephalosporins have a beta-lactam ring as shown in the bottom here, but attached to it, instead of a ring of 5 as noted here, you have actually a ring of 6. This is a cephalosporin formation, and then there's variable side groups attached to that. But you still have the beta-lactam ring, and it still does the same thing. It affects the integrity of the cell wall of the bacteria. These are examples of cephalosporins. Again, in the center you can see the beta-lactam ring in each one, and then the side groups. It's not important to memorize the side groups. It's important to understand that these are all beta-lactam drugs. They all affect the cell wall, and they all are created or synthesized to affect specific resistant bacteria. Kefzol in the upper left is what's called a first-generation cephalosporin. Just below it, clafuran or cefurotaxime. Below that, Fortaz or ceftazidime. And in the upper right, Rocephin or ceftriaxone are considered third-generation cephalosporins. Maxipine or cefipime. Below that is a fourth generation, and Tefloro, probably the most recent cephalosporin, is a 5th generation cephalosporin. Other beta-lactams include the carbapenems. Carbapenems, as you can see here, are 3 that we are going to discuss. They, again, all have the beta-lactam ring, but they have different side groups attached to them. Primaxin, which is also called imipenem, Merum, which is also called meropenem, and Invans, which is called ertapenem. These all are beta-lactam drugs devised in the lab for increased coverage of very resistant bacterial infections. And finally, there's a group of combination drugs where you take a beta-lactam antibiotic and you add another drug that's not an antibiotic but is an inhibitor of enzymes from the bacteria that fight antibiotics called beta-lactamases. And basically, if you have an inhibitor for beta-lactamase, you basically can inhibit that enzyme that the bacteria produce that basically disallows the antibiotic to work. The 2 of those that we use commonly are Unicin, which consists of an antibiotic, which is ampicillin, and a substance called sulbactam, which is a beta-lactamase inhibitor, which means it inhibits an enzyme called beta-lactamase that's produced by the bacteria that basically disrupts the ampicillin. Zosyn, being another commonly used beta-lactamase inhibitor combination drug, consists of 2 substances. One is piperacillin, which is an antibiotic, a beta-lactamase. You can see the beta-lactam ring. And tazobactam, which is a beta-lactamase inhibitor, which basically inhibits the enzyme that the bacteria make to basically disrupt or deactivate the antibiotic. So beta-lactamases are basically substances that are produced by the bacteria to disallow or disrupt the effectiveness of the antibiotic, which is why we have to keep developing more antibiotics, because bacteria, which divide very rapidly, develop resistance to antibiotics, and why one of the reasons that we're trying not to overuse antibiotics today, because of the risk of increased development of resistance to antibiotics due to the development of these substances like beta-lactamases that basically make antibiotics less effective. Many of these affect the beta-lactam ring. by producing substances called beta-lactamases. For example, penicillinase is a beta-lactamase produced by certain bacteria that basically takes the beta-lactam ring, as shown here on the left, and disrupts it, as seen here, into penicillic acid, which is now an ineffective substance and cannot disrupt the cell membrane. So the bacteria produce the penicillinase to fight against the antibiotic penicillin, and then we produce more complex antibiotics so that the penicillinase can't work on them, is how this occurs. Another way of inhibiting cell wall synthesis is by a substance called vancomycin. Vancomycin also inhibits the protein connections between the molecules of the cell wall. Vancomycin is a very complex molecule, but it's not a beta-lactam, does not have a beta-lactam ring, but it also works in a similar way. This is what vancomycin looks like. Its trade name is vancomycin. It's considered a glycopeptide, and it does act by binding to those protein connections of the beta-lactam ring and disrupting them so that when the bacteria tries to grow or tries to divide, the cell wall is disrupted and the bacteria dies. The next way in which antibiotics can act to affect bacteria is through action directly on the DNA. We said bacterial DNA is different than human DNA. It consists of one closed circular loop of DNA, and there are substances that have been devised to affect that closed circular DNA, but not affect our DNA. Those nucleic acid synthesis inhibitors include the fluoroquinolones, of which the examples we're using Levaquin and Cipro, or levofloxacin and ciprofloxacin, as well as a drug called Flagyl or metronidazole and a drug called rifampin. These drugs all act at the level of the DNA to inhibit the production of, or the duplication of DNA that occurs when the cell divides. If the cell can't divide, obviously you can't have bacterial infection, and your body's defense mechanisms can then break those cells down and destroy them. The quinolones, or the fluoroquinolones, such as ciprofloxacin and levofloxacin, act to block a group of enzymes that bacteria have called topoisomerases. Topoisomerases are enzymes that the bacteria have. If you think of the DNA as being like two ropes twisted around one another, and you hold one end and someone else holds the other end, and you try and separate those edges, if one person tries to separate them, they will get tied up in one another and you won't be able to do it. Topoisomerases basically clip the DNA so that it can unwrap, so that you can separate the two DNA strands, so that the DNA can make a copy of itself. If you block the ability of these topoisomerases to do that, then you inhibit the cell's ability to replicate its DNA, and therefore the cells cannot divide. Another drug called metronidazole or Flagyl is also an antibiotic. It's also used on certain protozoal infections. It also inhibits the synthesis of DNA, usually in bacteria that live in anaerobic or environments without air, and it basically disrupts the bacterial DNA so that it can't replicate itself. And finally, the third class or group of antibiotics that affect bacterial DNA are called the rifamycins. The classic example of that is a drug called rifampin. Rifampin, again, very complex molecule. It acts on bacterial DNA It actually inhibits the ability of that DNA to produce RNA molecules, which are going to direct protein development in that DNA. If that happens, then that bacteria cannot make any proteins, and if that bacteria cannot make proteins to allow it to survive, that bacteria will die. Rifampin is used very commonly in treatment of complex infections such as tuberculosis. The third bacterial process that is disrupted by types of bacteria. The next group of antibiotics does inhibit protein synthesis. It's listed here. We will talk about these briefly, but they all do the same thing. They all inhibit the ability of the bacteria to produce proteins necessary for the bacteria to survive, and therefore the bacteria will die. Remember, we said that bacterial ribosomes, which are the organelles that are responsible for protein synthesis, are different than our ribosomes. They consist of two ribosomal RNA subunits, a 30 and a 50, which is different than ours, which provides sites for antibiotics to be devised to affect these. And this shows how they do it. As we talk about these, again, take-home message is all of these antibiotics act to affect the function of the ribosomes, which is to produce proteins necessary for life for the bacteria. This is a list of the common antibiotics that we're going to talk about that do this. They're divided up into groups. We will talk about them by group as we go through. The first are the aminoglycosides. Aminoglycosides basically are complex, very older antibiotics. They're listed here. The most common one we will come across is gentamicin. Also, neomycin is a more topical antibiotic. They inhibit protein synthesis by binding to the 30S subunit, inhibiting the ability of proteins to be made by the ribosomes. The macrolides, of which the examples are erythromycin, azithromycin or zithromax, clarithromycin or biaxin, are all a group of complex molecules that inhibit protein synthesis by binding to the 50S subunit, again, inhibiting protein synthesis. And the bacteria cannot make proteins, and therefore the bacteria will die. In addition to these, several other antibiotics, such as the tetracyclines and the lincosamides or clindamycin or clindamycin, are important. Clindamycin is a very commonly used antibiotic to treat certain infections of skin, certain infections including methicillin-resistant staph infections. It binds to the 50S subunit also and inhibits bacterial protein synthesis. The tetracyclines, as you can see on the left here, actually bind to the 30S subunit, again inhibiting protein synthesis and the actual production of the proteins by the ribosome. The next activity that antibiotics work at is blocking folate biosynthesis. Again, we said that folate is a necessary substance for the production of DNA, for the production of certain proteins. And we said that while our cells can take in tetrahydrofolic acid or folic acid in our diets, a bacteria cannot. They have to produce tetrahydrofolic acid from various precursors, and we have devised antibiotics and substances that block several steps in those. Here is a chart showing the sulfonamides blocking the step early on in the production of dihydropteric acid, and then trimethoprim blocking the transfer from dihydrofolic acid to tetrahydrofolic acid. Both of these are necessary steps in bacteria to produce the folic acid to produce the DNA of the bacterial cell. If we block these steps, we block the production of DNA and the cell dies. This is an example of folate synthesis inhibitors. Again, the sulfonamide, sulfamethoxazole is the generic, Gantitol is the trade name, and then the antifolate agent trimethoprim or Primasol. Most of the time, these are combined together as a drug called Bactrim or Septra, which consists of trimethoprim sulfamethoxazole. So it blocks two steps in folate synthesis in bacteria. Again, not affecting our folate synthesis, since we can take in folate from our diet. And finally, we said that certain types of bacteria have an outer membrane on them, and there is an antibiotic called polymyxin D that can affect that outer membrane outside of the bacterial cell wall. We said that gram-negative bacteria, in addition to having a cell wall outside the cell membrane, have a membrane outside of that, as shown here on the diagram on the right. And if we can affect that outer membrane, we can disrupt that part of that cell wall. And antibiotics such as polymyxin B do that. It's mainly used as a topical antibiotic. It's a very commonly found antibiotic in topical antibiotics such as neosporin, polysporin, cordysporin, and it basically disrupts the outer membrane of gram-negative bacteria, obviously not affecting gram-positive bacteria at all, since they don't have an outer membrane. This is what polymyxin B looks like. Again, a very complex structure produced in the lab, not a naturally occurring substance. As you are transferring patients who may be on antibiotics, you have to understand that antibiotics, while directly not affecting our cells in the way they affect the bacterial cells, antibiotics can cause various types of adverse reactions, some of which do require EMS transfer, some of which can be very much life-threatening. We divide these down into three groups. We divide them down to what's called an intolerance, an allergy, and an interactions. Intolerance basically are annoyances that some antibiotics cause. These are not life-threatening in most cases. They may be a clue that a different antibiotic may be better, but they are not in any way affecting the antibiotic's effectiveness. They do not affect the ability of the antibiotic to do what it does, but they are annoyances, and these can be such things as dermatologic problems like rashes, which again don't provide any life-threatening condition, but certainly can be an annoyance. Many antibiotics do affect the gastrointestinal tract, causing nausea, vomiting, diarrhea. Again, if this is minor, this can be overcome. If this is more severe, it goes beyond an intolerance and into the realm of an allergy. Musculoskeletal, many antibiotics can cause cramping. These can be very mild. The fluoroquinolones are notorious for this, causing cramping and tendinitis conditions. However, this can actually progress to more severe conditions, including ruptured tendons and things like that. Also, some opportunistic infections can occur. Many people, when they take antibiotics, can develop secondary infections such as yeast infections because of the antibiotics' ability to clear out normal bacteria and allow other organisms that wouldn't normally grow to grow. Antibiotic allergies are more severe. These likely have more negative effects. These can be mild, such as hives or urticaria, more severe itching. They can be more severe nausea and vomiting, and they can continue to the point where they can lead to dehydration. They can lead to secondary infections like Clostridium difficile enterocolitis, which occurs when normal enteric bacteria in the gut are destroyed by antibiotics, allowing the abnormal bacteria to grow, and leading to the most severe part of that, which would be anaphylaxis, which is a life-threatening condition due to, in most cases, an allergic response to an antibiotic, which can lead to respiratory compromise, can lead to cardiac abnormalities, can lead to severe hypotension, shock, and death. Penicillin allergy is a very, very serious allergy. All of these antibiotics can, in many ways, affect people in a mild way to a life-threatening way. Antibiotics also can interact with other medication. There's many different medications that are affected by antibiotics. For example, many classes of antibiotics affect the effectiveness of contraceptive pills. Need to be aware of the fact that if you're taking a contraceptive pill and you're taking antibiotics, that antibiotic may decrease the effectiveness of that pill, making that pill not as protective as it should be. In addition, use of alcohol while taking antibiotics can increase side effects, particularly GI effects such as nausea, vomiting, diarrhea. In addition, certain antibiotics such as Bactrim or trimethoprim sulfamethoxazole and Flagyl or metronidazole can cause more severe reactions, severe abdominal pains, headaches, flushing, and other life-threatening reactions, period. Many classes of antibiotics increase the risk of bleeding. You'll transfer many patients that are on anticoagulants such as warfarin or Coumadin. People like that are on antibiotics are at increased risk of bleeding episodes, and it's an important question to ask when you're surveying patients with potentially life-threatening conditions, and you're surveying their medications, making sure that when you ask them, are you on any new medications, antibiotics could be a source of the problem that they're having. Aminoglycosides such as gentamicin can cause kidney damage, can cause hearing loss, especially if they're used with certain drugs, antifungal drugs, muscle relaxants, diuretics, and the like. And then the macrolides, the azithromycin, clarithromycin, and erythromycin have been known to cause cardiac dysrhythmias if taken with certain antihistamines and certain statins. These are all things to keep in mind as you're transporting patients with conditions such as abnormal rhythm function, abnormal cardiac activity, and when you're taking your history and you find out they're on an antibiotic, that may have something to do with their emergency medical condition. This is a chart showing the many of the common reactions and that you can have to certain antibiotics. This is not a complete list, but it certainly is gives you a good idea of the types of side effects you can get with the interactions between antibiotics and other drugs, or antibiotics and other antibiotics, and something to keep in mind as you're transporting patients. That ends this discussion of antibiotic use for EMS providers. Hope this was helpful.